Today's the day we talk about special sensory. We had previously discussed general sensory, which were body-wide. Special sensory, we had gave the designation that special senses are going to be located in the head only. So that kind of gives a clue to these special senses and at least where we're going to find those receptors. And if we go down the list of the 5 special senses that we have, we're going to see the first one, olfaction or smell, is going to be accomplished in the nasal cavity. So I'll just maybe make that note, nasal cavity. Then gustation or taste, this is going to be done by taste buds in the oral cavity. So we'll look at the receptors in the oral cavity responsible for taste or gustation. We looked last time into the eye, and we separated the eye into the 3 layers. There was the outer fibrous layer, the middle vascular layer, and that innermost neural layer that we described as the retina. We drew those cells of the retina, and that's what accomplished vision. I'm sure we'll review that again today. And then the last thing that we looked at the anatomy of last time, we looked at the anatomy of the ear, and way in the deep inner parts of the ear, we separated the ear into the external part, the middle part of the ear, and then the inner ear. I'm just going to list inner ear. And I'm going to point to both hearing and this equilibrium and balance. We're going to see this inner ear has the 2 different parts, the cochlea, that's responsible for hearing. Well, I'll just leave it as inner ear for now, and we'll see which parts of the inner ear are responsible for hearing and which parts of the inner ear are responsible for this equilibrium and balance. And we are starting with olfaction first. So you've got some questions on number 4. This is the bottom of the first page where we're talking about these olfactory receptors. And we're going to see these olfactory receptors are going to be embedded in a layer of epithelial tissue that's found in the superior nasal cavity. So if we go to this slide, it looks a lot like this large half-head model that I keep pointing to, because on the superior part of the nasal cavity in this half-head model, that's the only place where we can see those olfactory nerves. So olfactory epithelium, we can see this is a type of epithelial tissue that has embedded in it these olfactory nerves. So reading through number 4, it says olfactory receptors, we're going to see, are going to have cilia on them. If we can zoom in, here are those olfactory nerves, and they have cilia down here that are going to be able to detect odor molecules. Those are chemicals dissolved in air. So these olfactory receptors, you can see they're embedded in this mucus layer. That also helps trap these odor molecules. So olfactory receptors have cilia that detect odor molecules dissolved in solution. The olfactory epithelium, which is this layer of tissue right here, is the organ of smell and is located in the roof of the nasal cavity. That's why when you're trying to smell stuff, you try and take air into the nasal cavity very quickly. Going back to this picture, remember, if you breathe air in relatively slowly, the air is going to move in through the inferior and middle nasal meatuses. But if you want to smell things, you pull air swiftly into the nasal cavity, and that pulls it up to this superior nasal cavity where we have the olfactory epithelium. You think of smelling a flower. If we go back to this picture and look more closely at those cells, we can see those olfactory receptors, they're specialized bipolar cells. These are bipolar neurons. They've got the dendritic end on one side, the cell body is right in the middle, and then they have the axon portion that goes off in synapses with the postsynaptic neuron. There's a collection of postsynaptic neurons there in the olfactory bulb. So to read the second half of number 4, we have these olfactory receptors. They're specialized bipolar neurons. We can see they're called olfactory receptor cells or olfactory sensory neurons. So olfactory sensory neurons are embedded in the superior nasal cavity, and their axon portions we can see collectively go through the holes in the cribriform plate. That was our olfactory foramina. We can see that the axon portions of those olfactory receptor cells actually make up what we call the first cranial nerve, our olfactory nerve. So olfactory nerve leads into this olfactory bulb, and then we can't see it here, but if we were to follow that bulb back, it would narrow and become what we call the olfactory tract. Taste buds, we're going to call these gustatory epithelial cells. So in the oral cavity, I'm going to jump back a couple of slides and show you where these gustatory cells are scattered. When we look at one of these gustatory cells on the next slide, we're going to see that the apical surface of these gustatory cells, they have tiny hairs, little microvilli that we call gustatory hairs. Here's a picture of one of these. This is a taste bud. We can see it's made of several of these gustatory cells. And these gustatory cells, you can look at the apical surface of those gustatory cells, and they have those gustatory hairs. All of those gustatory hairs are facing this little tiny opening called a taste pore. And I'll go back a couple of slides to show you where these taste buds are scattered all over the oral cavity. But each taste bud we can see is a collection of these gustatory cells. And you'll notice that those gustatory cells are different shades. There's a lighter shade and a darker shade gustatory cell. Those gustatory hairs are the ones that have taste receptors on them. And it's interesting that these receptors, they change, as you age. And that's one of the reasons why the things that you find pleasing to eat might change as you age as well. But going back to this slide, it shows that each of those sensory neurons that are hooked up to taste buds, if it's a taste bud on the anterior 2/3 of the tongue, those sensory neurons are part of cranial nerve number 7, our facial nerve. We said on the posterior 1/3 of the tongue, that's where taste is picked up by cranial nerve number 9. That's our glossopharyngeal nerve. What you can also see here is that on the epiglottis, and even some of the throat there, the pharynx, there at the very back of the throat, there are some sensory neurons that are picking up taste that belong to the vagus nerve. I did not add the vagus nerve picking up taste on the epiglottis and the lower part of the pharynx in our cranial nerve sketch. But you have that box number 3, where anterior 2/3 of the tongue, that's cranial nerve number 7. Posterior 1/3 of the tongue, that's cranial nerve number 9. And then on the epiglottis or the lower pharynx, this is the vagus nerve. The slide that I kept skipping over is this one, and it gives you the answer to question number 2. Those sensory neurons that we just found on the surface of the tongue, they're picking up different types of tastes. And there are 5 different classes of tastes that can be picked up by these gustatory cells. In addition to the 4 common distinct taste sensations like sweet, sour, salty, and bitter, there is a 5th taste called umami, which is associated with the taste of biting into certain proteins, like steak or chicken or fish, or biting into like a big portobello mushroom or something like that. Those taste sensations are not sweet, they're not sour, they're not salty, they're not bitter. They're collectively belonging to a different group that we call umami. So 5 distinct taste sensations. Those taste sensations are being picked up by one of these sensory neurons, either on the tongue or on the epiglottis. So next one on the list is looking at vision. And we saw some of this last time because we were looking at structures of the eye. When we were listing the 3 layers of the eye, there was the outermost layer that we called the fibrous tunic. I'll say fibrous layer or fibrous tunic. There were 2 structures in this fibrous layer that we had to know: the sclera. This was the white of the eye. And the cornea. This was the clear anterior part of the eye. Then as we went through this outer fibrous layer, that took us into the second layer that we called the vascular layer, vascular tunic. Some textbooks will call it a tunic rather than a layer, so let's just list that. The key parts to the vascular layer or vascular tunic that we had to know there was the choroid. This choroid layer is where we found a bunch of blood vessels. There were blood vessels, there were nerves. If we followed that choroid layer around to the anterior side, it led into this kind of structure that we called the ciliary body. Is that right? Yeah, ciliary body. I said this ciliary body we would subdivide into two structures, what we called the ciliary process and the ciliary muscle. So the ciliary muscle is going to allow the pupil to constrict or dilate. This ciliary process. If we looked at the ciliary process, kind of sticking off the ciliary process, we found those suspensory ligaments. These suspensory ligaments, they held the lens in place. And they are stretching directly off of that ciliary process. If we looked on the anterior side of this ciliary body, we would find the colored portion of the eye, the iris. And I guess we could list the pupil. On the last thing to put on here, the third innermost layer of the eye, we call this the sensory layer or neural tunic. This sensory layer, there's really two things that we needed to be able to distinguish in the sensory layer. This innermost layer was made up of a pigmented layer. These cells were like shiny mirrors, and on top of the shiny mirrors there was the retina. The retina, technically, there were 5 cells that make up this retina that we need to know. In fact, I'm just going to put a little star next to that one. I've got the 3 layers. Let's see if we can find these. And we'll talk a little bit more about the physiology of the eye. What is the eye trying to accomplish? We saw the extrinsic eye muscles, these muscles that help move around the eye. And we listed them on the lab list. Looking at the slide that looks like this, it is the best representative of the model that we will see where I'm likely to pin these different types of muscles. And as far as the rectus muscles along the eye go, those are easy to distinguish. The superior rectus, obviously that's on the superior part of the eye. Inferior rectus is on the inferior part of the eye. But then maybe a little more difficult, especially if there's just this single eyeball sitting here looking at you, you need to be able to tell medial from lateral. So how we can tell medial rectus from the lateral rectus is that it's always the lateral rectus. If we look in between the lateral rectus and the inferior rectus, that's where we find this muscle called the inferior oblique. So inferior oblique tells us lateral, and it should be easy to find the superior oblique. It's on the top of the eye, and it's coming in at an oblique angle. We found those on the models last time, but I think in your lecture handout it wants you to know which direction these muscles are going to pull the eye. And these arrows, I think they do somewhat of a decent job of showing the direction that these muscles are pulling on the eye. But I don't like the way that these are showing the oblique muscles and the direction of this. I found a better picture. Just before I switch to that better picture, let me show you these rectus muscles. You can see if this rectus muscle, this medial rectus, if it were to contract or shorten, it's going to pull the eye towards the midline of the body. The lateral rectus, you can imagine if the lateral rectus were to shorten or constrict, it would pull the eye away from the midline. It would be you kind of looking in the lateral direction. The superior and inferior oblique, look how if the superior, I just said superior oblique. If this superior rectus were to constrict, notice how it's going to pull the eye up, but also towards the midline. That's up and towards the midline a little bit, just like the inferior rectus. If this was to shorten or contract, it would pull the eye inferior, but also towards the midline a little bit. Let me go to the next slide to show you the same things about the rectus muscles that we just talked about. This is kind of a confusing looking picture because it's showing us both eyes at the same time. We've got the right and the left eye, but instead of really focusing on the eyeballs themselves, let's look at this represents the medial rectus. You can see whether we're talking about the right eye or the left eye, if either of those eyes were to contract their medial rectus, it would cause both of those eyes to move towards the midline of the body. So crossing your eyes, this would be those medial rectus muscles, both pulling the eyes towards the midline. You can see lateral rectus. Both of those lateral rectuses are going to cause these eyes to move laterally, away from the midline. You can see that inferior rectus pulls the eye down and towards the midline. Both inferior rectuses are going to pull their eyes down and towards the midline. Both superior rectuses are going to pull the eye up and towards the midline. It's only the obliques, the superior oblique and inferior oblique, that are going to pull the eye either up or down in a lateral fashion. So superior oblique, you can see both superior obliques are going to cause the eye to look down and laterally. Superior oblique, eyes move down and laterally. Inferior oblique, you can see these are going to cause the eye to move up and laterally. This picture did not convey that very clearly, so luckily I was able to find this picture that does a great job of showing us the direction that each of those extrinsic eye muscles are going to cause the eye to move. That is, I know we kind of jumped around a little bit, that is, where is that? Thank you, I knew it was on there somewhere. Yeah, the first part of the third page, question 21 through 26. Did everybody get that?
Can we just go over it?
I think we can. So when you're looking through all of that, yeah, go ahead and take that picture too if you want. But what this picture should help us do is walk through those questions, 21 through 26. So in 21 it says it turns the eye up and towards the midline. So the ones that are going up and towards the midline here is the superior rectus. Up and towards the midline is superior rectus. Question number 22, it says turns the eye down and towards the midline. So down and towards the midline, this is the inferior rectus. Then in question 23, it says, turns the eye down and laterally. So we're going down and laterally. The only muscle that's moving these eyes down and to the lateral direction is the superior obliques. Now moving to 24, it says turn the eye away from the midline. So it looks like the one that's just going away from the midline is the lateral rectus. Not up or down, just away from that midline. That would be lateral rectus for 24. Then in 25, turns the eye up and laterally. So the only one here that's making the eye go up and also to the lateral side is our inferior oblique. And then the last one on 26 just turns the eye towards the midline, and this is medial rectus. So confusing diagram there, but it is helpful when you think about which way those extrinsic eye muscles are pulling on the eye. Okay, so extrinsic eye muscles crossed off the list. We now get into those layers of the eye that we listed on the board. There's the outer fibrous layer, there's the second vascular layer, and then the innermost sensory layer. If we look closely at those individual layers, we have listed these on the board. So we know the sclera is the white part of the eye, the cornea is the clear anterior part where you would stick a contact lens. When we get into that vascular layer, there is the choroid layer with the blood vessels, the iris, the colored portion of the eye, the ciliary body, which we said was made up of the ciliary process, supporting those suspensory ligaments and the lens, and then the ciliary muscle that does this constriction or dilation of the pupil. And once again, we'll look at the sensory layer, at that shiny pigmented layer and the 5 cells that make up the retina. But first, to this fibrous layer, the outermost fibrous layer of the eye starts with this sclera, the white portion of the eye. As we get to the anterior part of the eye, that white sclera gives way to this clear cornea. So the cornea was this clear anterior part, and then you've got the rest of the sclera, the white portion of the eye. The vascular layer, let's just stick to this slide and look at the vascular layers. Everything in red, so we can see the sclera is the fibrous layer, the red is part of this vascular choroid layer, and then the yellow is part of that sensory layer, the retina and pigmented layer. But if we follow this choroid layer away from the back side of the eye and towards the anterior part of the eye, right there, and here's where I zoom in on it a little bit, you can see that there is an expansion of this choroid layer. It expands into what we call the ciliary body. This ciliary body is 2 things. There is the ciliary process, the little part of the ciliary body that the suspensory ligaments grow off of. And here you can see those suspensory ligaments holding onto the lens. Kind of just underneath the ciliary process, in the middle of that ciliary body, there are these ciliary muscles, which are going to cause, like we said, a constriction or a dilation of the iris itself. This looks like a brown iris, and we can see either constricting or dilating this iris is going to cause the size of that pupil to change and adjust how much light hits that lens. Eventually, we're going to get into this yellow retina layer, and we're going to see, like we said, the 5 cells of the retina and the pigmented layer that they sit on. But let's jump past that to this that actually shows us a picture of the retina. This is the pigmented layer of cells that act like shiny mirrors, and then we can see those 5 layers of neurons that make up the retina. So together, this whole thing is our sensory layer of the eye. And it was a little counterintuitive as to what these arrows were pointing to, but remember, this entire chain of neurons, none of those neurons are capable of detecting photons of light except for the dendritic ends of these photoreceptors. So this pathway shows as light initially comes into the eye, we said it has to bypass it. It moves through these ganglion cells, these bipolar cells, and even the axon and cell bodies of these photoreceptors. It's not until the light bounces off and is reflected away from this pigmented layer that those photons ever trigger the dendritic ends of these photoreceptors. It's only the dendritic ends of these photoreceptors that can send signals to the bipolar cells and to the ganglion cells. So again, light has to come through and bounce off of this pigmented layer, and then it's picked up by these photoreceptors. We listed, when we were talking about the different types of photoreceptors, we can see that light, as it comes into the center of the eye, is hitting the back portion of the eye in an area that we called the fovea centralis. This is where we found the highest concentration of cones. We said there were rods scattered around the perimeter, and then right there where the optic nerve comes into the back of the eye, we said there were no rods or cones. You needed rods or cones to form an image, and the lack of rods and cones in this optic disc prevents us from forming an image. So optic disc is sometimes referred to as the blind spot. It's where that optic nerve leads into the back of the eye. And since we can't form an image there, you can see that that optic nerve doesn't come in directly into the back of the eye. It's kind of off-center a little bit. What is directly center in the back of the eye is that fovea centralis, that highest concentration of cones. We said cones, if we want to look at this fovea centralis again, those are the photoreceptors, and there are going to be more cones in this macula lutea than there are going to be rods. I thought I had a picture of a rod and cone. I'll just go back to this one. Since we were talking about rods and cones, we compared rods and cones last time briefly. If we want to continue to answer those questions on the lecture handout, there's some comparison about rods and cones. we are going to see that cones are going to be used for color vision. Cones are going to be used in the daytime, so bright light. If it's bright, you're using cones. If you're trying to pick colors out, you're using cones. And if you're trying to see sharp edges on something, like one of those visual acuity charts, you're using cones. So high visual acuity, color vision, and vision in bright lights all respond to cones. Rods, on the other hand, are going to be what you would use in dim light. And just think about that. Think if we're walking around that field at night, we're going to see things in shades of gray. We're not going to see colors as well. In addition to that, it's going to be hard to see sharp edges. We might see the side of a barn or something like that, but the sharp edges, it would be hard to do a visual acuity chart in the dark. We said that you are also more sensitive to light in your peripheral vision. So there are more rods out there in the periphery of your retina. We drew this last time. I don't know if it's worth doing it again. Maybe in our lab review time, I'll put this sketch on the board again where we drew the pigmented layer and then we drew the photoreceptors that were talking to these bipolar cells. Bipolar cells were talking to ganglion cells. And then in between the photoreceptors and bipolar cells, we had these horizontal cells that really just adjusted the sensitivity of those bipolar cells. And just like those horizontal cells were adjusting the sensitivity of bipolar cells, these amacrine cells are adjusting the sensitivity to those ganglion cells. We've zoomed out away from the retina and we can see this optic nerve as it leaves the back of the eye. And if we're on our lab list, you can see the optic nerve is, the portion of the neuron that leaves the back of the eye. But then right after it leaves the eye, we can see both of those optic nerves cross one another. This is the optic chiasma. And notably, only those medial fibers actually cross over. The lateral fibers stay on their own respective sides. But you have optic nerve that leads to the optic chiasma, and you can tell whether you're supposed to write optic chiasma because it says just to the right of that, axons from the medial half of each retina cross to the opposite sides of the brain, right there in the optic chiasma. After that chiasma, after the cross occurs, we can see the rest of the neuron is going to be referred to as the optic tract. And I think the word tract is given to you there, so you just have to write the word optic. And I did this before when I was using my hands. I said my hands are the optic. If my fists are the eyeballs, my wrists would be the optic nerves, and then where my forearms are crossing is the optic chiasma, and then my elbows would be the optic tracts after the cross. But maybe after that, if my elbows are the optic tracts, then my shoulders here are, if we follow these optic tracts back a little further, we can see that each of those optic tracts are dead-ending into this structure. I wish I could zoom in on it a little more, but you can see there's a collection of neuron cell bodies. So there's a synapse between these two different neurons. This area is called the lateral geniculate nucleus of the thalamus. I think part of that is given for you there. The nucleus of the thalamus is given. You just have to write lateral geniculate nucleus of the thalamus. So here's a slide that we did see last time, and what we know now is that just behind that pupil sits a lens. And if the pupil, here's a normal size of our pupil, if we were to constrict the pupils, we know during rest and digest there is a constriction of the pupils. This parasympathetic stimulus causes those smooth muscles to constrict, and now the diameter of that pupil is not quite as large versus the sympathetic division. When the sympathetic stimulus causes these ciliary muscles to relax, it dilates the pupil. What we can't see on here, but I'm about to switch to, just on the back side, the other side of this pupil, there are going to be suspensory ligaments that hold that lens in place. There again is our lens. We can see the green suspensory ligaments that are kind of like the springs on a trampoline. They're holding that lens in place. The lens is the trampoline in this analogy. And you can see that when the sympathetic nervous system causes the pupils to dilate, that stretches the springs on that trampoline very tight. So the suspensory ligaments get drawn very tight. And what that does is it stretches out the lens. The lens becomes flattened and weaker. This is actually going to be good for accommodating far vision. So if you're in fight or flight, right, and we hear some kind of stressful noises down the hallway, our eyes are immediately going to try and look for, how are we going to get out of this situation? So accommodating for far-sighted, for far vision, the lens needs to become flattened. And what stretches and flattens out that lens is by the pupil dilating. Compare that. So sympathetic stimulus causes a stretching of those suspensory ligaments and a flattening out of the lens. Compared to parasympathetic stimulus, here's that same diagram, but you can see in parasympathetic stimulus it causes those pupils to constrict. And now that those pupils are not quite as big, there's nothing stretching those suspensory ligaments. Now there's actually slack in those suspensory ligaments. And so without anything stretching on that lens, the lens kind of recoils and it becomes more of this rounded shape. If I go back and forth between these two slides, look at how the lens is more rounded under parasympathetic stimulus and slightly flatter during sympathetic stimulus. It's not a lot, but you can see rounded, parasympathetic, flat is sympathetic. Rounded is for near vision. Remember, in Rest and Digest, you're reading a book on the couch. That book is right in front of your face. So accommodating for near vision, the lens becomes rounded. And if we're trying to accommodate for far vision, the lens becomes flattened. I want to draw some other things on the board that help us think about the inner ear, because there's a lot going on there. We've got hearing and we've got equilibrium and balance all going on in that inner ear. So before we get to this inner ear, let's remind ourselves of the structures of the external and middle ear. We start with the external ear. We said the auricle or the pinna was the earlobe, and its job was to catch those sound waves and funnel them into this external acoustic meatus, the ear hole or the ear canal. We said that by having those different shapes and bumps and ridges in the auricle of the ear, it allows it to capture sound waves coming from different directions and funnel them into the ear. We said it sounds different from your perspective if I'm trying to communicate to you standing in front of you than if I was standing behind you. You would still be able to hear me, but it would sound different to your ears because those sound waves they're bouncing around that auricle and being funneled into the ear a little bit differently. So it tells us something about the direction of those sound waves. Once sound waves enter this external acoustic meatus, they're going to end up slamming against our eardrum, what we call the tympanic membrane. And it's really that tympanic membrane that is the dividing line between the external ear and the middle ear. The middle ear, we really said it was the tympanic membrane, these three delicate inner ear bones, as well as this auditory tube or eustachian tube. This connects the middle ear to the back of the throat. So we said if you are flying in an airplane or riding up and down mountains, you sometimes feel your ears pop, and that's just the pressure equalizing on either side of your eardrum. So these auditory tubes, they open to the back of the throat, so sometimes like a deep yawn or chewing gum can help allow the air pressure to equalize on either side of that eardrum. But if we zoom into this middle ear, you can see those three ear ossicles that I pointed to. The one that actually touches the eardrum itself, or this tympanic membrane, that particular bone we call the malleus. We said the middle ear bone is the incus. For some reason the S got cut off there, but incus. And then stapes is this stirrup-shaped bone. What was noteworthy about the stapes in this picture, it's covering up the oval window. So I asked you to put some kind of note somewhere that the stapes, if it's present, it's covering up that oval window. There was one of these large inner ear models, looks like I'm using both of them on the practical, But if I flip this inner ear model over, let's just take it out of this. This is the only place that I can show you an oval window where there is no stapes. So the stapes has been removed to show us this oval window, as well as a little round window, which we can see indicated by that little dot. We can't see the oval window, but we can see the round window. And on this model of the inner ear, they're right next to one another. More on the oval and round window in a second. But we're going to jump past that into this inner ear. But I guess right before we do jump into this inner ear, there's something that I wanted to mention about these muscles that attach to those inner ear bones. There is this stapedius muscle. It's a tiny muscle that connects to the stapes. And then there's this tensor tympani muscle that connects to the malleus. These are skeletal muscles whose job it is, if these muscles were to constrict, what they're trying to do is they're trying to bear down on these ear ossicles and prevent them from vibrating. Remember, we said it was the job, and we'll say this again by the end of this lecture, it's going to be the job of these delicate ear bones to take these the vibrations that hit the tympanic membrane. They have to take those vibrations and amplify them as they're sent into this inner ear. So I used the scenario of there was a guy in my class years ago that worked at the airport where he was around airplanes that were landing all the time and really loud noises. If you're in areas like that, or even worse than that, there are some, like it's been a torture technique before, to blast sounds at people. And sounds, if they're above a certain decibel level, they can do damage to the eardrum and be very painful. And if maybe a minor example of that would be, it happens sometimes when I grab one of these models and rake it across the table, it makes that loud screeching noise. And usually if that hurts your ears, that muscle contraction that you feel in your ears are these muscles, the stapedius muscle and this tensor tympani muscle. Those muscles contract and they try and prevent these ear ossicles from vibrating. Too much vibrations could rip open holes in that eardrum. And so if vibrations get to this crucial level, then those two middle ear muscles, stapedius and tensor tympani, try and prevent those ear ossicles from vibrating to the point that they're going to cause damage. So again, I won't try and do that on purpose, but if I accidentally make one of those loud noises with the model, those are the muscles that you feel in your middle ear that contract. Okay. Now to the inner ear, and to do this, I think maybe one of the best things is to just make another chart on the board. So just a reminder, we're talking about this inner ear, and when we're thinking about the inner ear, we can split it into this bony labyrinth. I'm going to put next to the bony labyrinth, we're going to put the membranous labyrinth. And inside the membranous labyrinth, let's just put the receptor region. We'll start off in this bony labyrinth. So we're looking at the outer candy shell that's made out of compact bone. And we said there were three parts of this outer portion of the inner ear. There was the vestibule. There was the semicircular canals. And then there was what we call the cochlea, the snail shell looking structure. Okay, Inside each of these structures, we're going to find these membranous regions, and I feel like we had a picture of that. So here's the inner ear. We can see this coiled snail shell that we're going to call a cochlea, and then we can see part of this vestibule. It has semicircular canals attached to it. If I can pull that inner ear out all by itself, here's where we can see this cochlea. We can see our vestibule. We can see these semicircular canals. But you can see in blue here, those blue structures in the middle of the semicircular canals, we're going to refer to those as semicircular ducts. I'm just going to list it on the board real quick. Semicircular ducts are found inside semicircular canals. And if you look inside the vestibule, there are, I guess we'll just go through in the same order as the slides. We can see those semicircular ducts in the middle of those semicircular canals. And I'm about to point to these gelatinous patches that you find inside the vestibule. Here again is the vestibular system. You can see that external kind of thin layer of compact bone that makes up the vestibule. We can see these semicircular canals that are anchored to that vestibule. And then now inside that vestibule, we're going to see these two gelatinous regions that have receptors on them. You can see those purple receptors. These two little gelatinous regions inside the vestibule, one is called the saccule and the other one is called the utricle. So let's put saccule. Saccule and utricle are going to be found within the membranous regions inside the vestibule, those two membrane patches that you find. We can see that this utricle is going to be closer to the semicircular canals. Maybe think the U in utricle tells you semicircular canals. I don't know. That might work. The saccule is closer to the cochlea. Both saccule and utricle, as you can see in this picture, both utricle and saccule, they contain a receptor region that we call a maculae. Let me write that in a different color. The receptor regions that we find inside saccule and utricle are called maculae. I'll maybe list what this maculae is a receptor region for, since I have a little bit of space. Maculae are going to be responsible for what we called static equilibrium. I'll give an example of that. I'm going to write static equilibrium. An example of static equilibrium, this would be linear acceleration and gravity. So these maculae, they work when you are able to tell whether there is linear acceleration. So again, this example is you're in the car, your eyes are closed, but you could tell if somebody's slamming on the gas or slamming on the brakes. You can detect that linear acceleration through these maculae. Maybe the gravity example is, again, I grew up in a small town where I saw these carnival rides come to town and not get put together properly, so it scared me to the point where I could never do any of those big roller coasters. But there's a ride at Six Flags. I don't even know what it's called, but you go to the top of this tower, and then it spits you off the front of the tower, and then you just fall for like stories. Has anybody ever done that scary thing? I wouldn't do it either. But what the sensation that you're feeling there whenever you're falling. You can tell whether the elevator is going up or whether the elevator is going down, once again, through this maculae. So linear acceleration and gravity. Your body is still, so this is why it's static equilibrium. I'm just standing in the elevator, or I'm just sitting in the car, and I can tell whether we're going forward or backward, or if the elevator is going up and down. Static equilibrium. We're going to compare that to dynamic equilibrium in a second. And we said dynamic equilibrium was when your body was turning through the air and you had to be able to know when to come out of this somersault to land on your feet. So those are different receptors in a different region. But if we're thinking about just static equilibrium, this is monitored by maculae. These are receptors found in the saccule and utricle, which are compartments of the vestibule. Here we are looking at them a little more closely. Here are those receptor regions, the macula of both the utricle and saccule. And here's where I've kind of run out of room to add to some detail about these receptor regions. So one of the things that I'll just put that's noteworthy about these receptor regions is that every one of these receptor regions, whether it's the maculae or whether it's these other two specific receptors we're going to talk about in the other columns, they all use hair cells. And we talked about hair cells before. These are specialized neurons that are capable of sending signals if they're stimulated in the right way. We won't have to draw hair cells, but you can see a few of them in this picture. They're hooked up at one end to a sensory neuron, and then the opposite end they have these stair-step-like cilia called stereocilia. If we zoom into those stereocilia, you can see they look kind of like this, where it's a stair-step all the way down. You know, on one side there's the longest cilia, and on the other opposite side there's the shortest cilia. And all of these stereocilia are embedded into some type of endolymph. So this picture shows all of that endolymph that each of these stereocilia are embedded in. And we know that how these hair cells work, if you were to bend the hairs in a particular direction, that might allow positive ions to rush into the cell. Something that was a side note but noteworthy, in endolymph we said there was a very high concentration of potassium ions. It basically is playing the role that sodium was playing when we were talking about neurons depolarizing. In hair cells, it's potassium that rushes in to cause the depolarization. But what we really need to know is if we look at those hair cells more closely, here's a picture of one of those hair cells a little bit more closely. I tried to sketch something like this a couple of days ago where each of those stereocilia, if we could look close enough at them, we would see that they're lined by these mechanically gated channels. It's a channel, but it's not an open channel. It's got a little gate there. So we can see the gated channel, and this gated channel opens and closes physically. And you can see the little titin protein that is connecting two of these mechanically gated channels. Since each of these mechanically gated channels are connected by that titin protein, you can imagine if we were to bend all of these hair cells towards that tallest cilia, what this is going to do is it's going to pull tight each of those little titin coils, and it's going to physically open up each of those potassium mechanically gated channels. So potassium rushes into this cell, it causes a depolarization. If instead those stereocilia are bent in the opposite direction where it adds slack to each of those titin proteins, then this is like physically slamming shut the door, so there is going to be no potassium that moves in. So if we bend the hair cells in one way, this is an inhibitory way of shutting off those hair cells, versus if the hairs were to bend towards the tallest stereocilia, this is going to cause those mechanically gated channels to open and send a signal to the sensory neuron. I only point this out because if we know how hair cells work in one place, like the maculae, we know how the hair cells work in all of the other places where we see hair cells. Anywhere where we see purple regions, that's where we find hair cells. We've crossed off the hair cells in these two maculae, collections of hair cells in the utricle or saccule. We said this was for our static equilibrium. So now let's move, while we're still over here in the vestibule region, let's move into the semicircular ducts, because I had pointed out those earlier. We can see the semicircular canals are the outer bony regions. They house those semicircular ducts, which we can see in blue. And as we approach the terminal ends of these semicircular ducts, we can see there's an expanded region called an ampulla. And inside those ampulla are where we find these receptor regions called the crista ampullaris. So crista ampullaris, these are receptor regions for what we're going to call dynamic equilibrium. So dynamic equilibrium, you can think of, instead of linear acceleration in gravity, this is telling you something about rotational direction. So if you're doing somersaults in the air and you're trying to land on your feet, or if I used to like jumping off of diving boards and doing tricks and landing into the water, but you've got to be able to tell where you are in these rotations. So the crista ampullaris is the receptor regions that help monitor this dynamic equilibrium. If we look closely at that crista ampullaris region here in this ampulla, there is the crista ampullaris. It's once again, you can see there's this gelatinous endolymph. And there are also hair cells embedded into that endolymph. And we know how hair cells work. If they were to be bent in a direction towards that tallest stereocilia, that's going to cause them to depolarize and send signals to the sensory neurons. But if they were to bend in the towards the smallest or the shortest stereocilia, this would be an inhibitory signal. So once again, hair cells behaving in the same way as they did in the maculae. But here they're detecting that rotational direction. So we've seen the crista ampullaris, we've seen these maculae. Let's now look at this purple, kind of a spiral-shaped receptor region that goes through the cochlea. We've made it down here to the cochlea, so that's the hard outer shell of this snail. If we were to look inside that, there is going to be a membranous region inside the cochlea. That's the blue structure that is on this slide called the cochlear duct. Let's list our cochlear duct. Inside this cochlear duct, the receptor region that we're going to have to pay attention to is called the organ of Corti. Organ of Corti, this is a receptor region that's just responsible for hearing. So hopefully we've got that. If we go back to this picture, that coil of bone that I keep describing as a snail shell is correctly referred to as the modiolus. And we can see it has an apex at one end. And if we look within this modiolus, we're going to see the organ of Corti, this what looks like a spiraled staircase that spirals around this modiolus. So here we are back at this coiled snail shell or a modiolus. We can see the apex at one area, and then we can kind of see the base of this modiolus with the cochlear nerve entering. We said this was like a coiled garden hose where there was a big hollow tube in the center. But this hollow tube has been subdivided into 3 different compartments. So we zoomed into just one of these hollow tubes, and we were wanting to list these 3 different compartments. Okay, I'll try not to look at that, but it's the same thing that I'm about to try and draw. I'm just going to come in here and grab a section of this that I'm going to make look like a wedge with soft edges. And then I'm going to kind of make a half circle above it and one of these half circles below it. Good so far? We're going to label these things our separate ducts. The one in the middle is going to be our cochlear duct. Above the cochlear duct is what we're going to call our vestibular duct. And the one beneath is our tympanic duct. There's 3 membranes and there's 3 ducts that we need to know. We've got the 3 ducts, so let's put the 3 membranes. There is between the vestibular duct and the cochlear duct. I'll put it in a dark blue color. Between vestibular duct and cochlear duct, there is this blue membrane that we're going to call the vestibular membrane. And then between the cochlear duct and the tympanic duct, there is another membrane. I'll put it in a light blue. This one is going to be what we call the basilar membrane. So basilar membrane. And there's only one other membrane that we need to add to that. And that is a membrane that looks, it's in the cochlear duct and it's attached to this little hinge kind of in the narrow part of the cochlear duct. So I'm going to draw this coming out and then kind of going right back to that narrow little hinge. This purple membrane is our tectorial membrane. So tectorial membrane. You can remember the tectorial membrane because this tectorial membrane sits directly on top of, let's put it in a brown color, these hair cells. Remember, there are 3 of these outer hair cells and 1 inner hair cell. Just list those things as hair cells. Those hair cells are located between the tectorial membrane and they sit on top of the basilar membrane. So under the tectorial membrane and on top of the basilar membrane. Maybe I can use this green color to show that there is a sensory neuron that hooks up to each of those hair cells and then makes its way out of the kind of the narrow end of this cochlear duct. It leads to this is a sensory neuron that leads to the spiral ganglia. Well, there's our sensory nerve going to the spiral ganglia. Okay, so we've got our 3 ducts, we've got our 3 membranes. So everything is going fine so far, but let's add to this. Am I doing okay so far? Let me know if there's questions. If I don't see it, just throw it out there. I'm going to use the orange and the green color to add something to the picture here. It's where we're going in our slides. The vestibular duct and this tympanic duct, these are going to be filled with a fluid called perilymph. I'll just maybe list it in here. So perilymph is going to be filling this vestibular duct and the tympanic duct. But if we look at the cochlear duct, it's going to be filled with a different fluid, what we're going to call endolymph. Hopefully that's easy. Endolymph. So I'm just going to maybe put the orange color in here to show that this entire chamber is filled with endolymph, just like these chambers are going to be filled with perilymph. Okay, so we've got our cochlear duct, this membrane-lined chamber in the middle, and the structure that we consider the organ of Corti is this thing right here. Organ of Corti, on your lab list, it's going to include those hair cells. It's going to include this basilar membrane that the hair cells are sitting on. It's going to include this tectorial membrane that is right on top of the hair cells. So any of those 3 things would be fair game for me to stick a pin in. If we can trigger the tympanic membrane to slam down on top of the hairs of the hair cell, that's going to be picked up as sound by the sensory neurons. We've set the anatomy up. Let's now add a little bit more to this. Are we feeling okay so far still? What I'm about to get to is how those hair cells get stimulated. How does this tectorial membrane move around? And we'll see a video, but I'll try and first explain this just with words. And we need to add something to this diagram. Let me catch up with some slides too. As I walk one direction, my pointer is on the other side, so let me go back over here. All we've done is zoom into this organ of corti. We can see this tectorial membrane, we can see our hair cells, we can see the sensory neuron, and we can see this basilar membrane that the hair cells are sitting directly on top of. I'm about to try and describe these mechanics of hearing. It starts basically by me causing these sound waves to vibrate through the air. And these sound waves, we know as they're moving through the air, your auricle is catching these sound waves and funneling them into that external acoustic meatus. That's causing the tympanic membrane to vibrate back and forth. And what I haven't drawn on here, we have to imagine that tympanic membrane vibrating. As that tympanic membrane vibrates, it causes each of those ear ossicles to vibrate. And there was that final ear ossicle that we called the stapes. We said the stapes was always sitting on top of something that we called the oval window. The oval window now has been added to this diagram, and you can imagine anywhere on these inner ear modules, if you can remove the stapes, it would reveal this oval window. And if we could somehow dive down through that oval window, we would end up in this vestibular duct. We would notice that the chamber is filled with this gelatinous fluid called perilymph. So it's just jelly-filled chamber. And that's why if, the stapes, if it's floating right on top of this perilymph, if we were to vibrate the stapes, what it does, let me grab a different color here. Vibrations from the stapes are going to send pressure waves out in all directions from this oval window. So this is where that analogy of trying to get the water on the surface of a swimming pool to become very rocky. One way you can do that was taking like an inner tube and submerging it into the water a little bit and letting it rebound to the top of the water. And when you're doing that, if you can maybe imagine doing that, it's sending out these pressure waves in all directions away from the inner tube. It's like what happens when the stapes vibrates. And you can see if these vibrations hit against the wall of the vestibular duct, then they bounce off. They're going to bounce off and come back this way. These are coming up, bouncing off and coming back this way. These waves are going directly down to this vestibular membrane. Once the stapes vibrates enough and sends pressure waves through the vestibular duct, those pressure waves, once this perilymph is rocking back and forth, it's going to put pressure on this vestibular membrane. It's going to cause the vestibular membrane to move a little bit. And when the vestibular membrane moves a little bit, it's going to start to disturb that fluid in the endolymph. And as these pressure waves move into the endolymph, if they're strong enough, they're going to slam into that tectorial membrane. And if that tectorial membrane wiggles a little bit, then it's going to be picked up by, it's stimulating these hair cells. And we've got a signal that's sent to the brain. So this is where the sound waves are picked up, but it's not where the pressure waves stop. You might be surprised to learn that these pressure waves, as this endolymph is rocking back and forth, and our tectorial membrane starts to rock back and forth, that disturbs this basilar membrane. So this basilar membrane down here also starts to vibrate, and these pressure waves continue on into our tympanic duct. They actually would continue to bounce around and bounce off the walls of this tympanic duct. They would actually bounce back into the basilar membrane, and we would have this echo effect, a constant echo effect in the brain if these pressure waves couldn't get out somewhere. We see that the pressure waves start in the oval window, and they're actually going to leave. The way out is through the round window. So the round window is what allows these pressure waves to actually leave the inner ear. It's what prevents you from hearing a constant echo from whatever the first thing I said today was. So pressure waves start in the oval window, they end in the round window. And somewhere in the middle of there is when they were triggering those hair cells and being picked up by the brain. So I want to talk about the different types of signals being sent to the brain and whether they're perceived as different pitches, like different notes, or different amplitudes of sound, whether it's loud or whether it's quiet. I think I have a picture of exactly what I was trying to describe here. We've got the sound waves actually slamming against that tympanic membrane. That causes those ear ossicles to amplify those vibrations. And right there you can see as the stapes vibrates, it's vibrating all of that gelatinous material underneath. They're using these blue lines to show the pressure waves that start to radiate from the stapes. So right there at the oval window, these pressure waves start to make their way through this vestibular duct. Interestingly enough, if they're a really high-pitched note, then they're not going to make their way very far into this vestibular duct. They're going to move across this vestibular duct into the cochlear duct. A really low-pitched sound is going to make its way further into this vestibular duct. I'll distinguish those in just a second with a little video. But pressure waves originate under this oval window. They start by spreading through this vestibular duct, and then at some point, those pressure waves are going to disturb this vestibular membrane. If the vestibular membrane gets disturbed, you can see how that moving vestibular membrane is going to disturb the endolymph. And if this endolymph gets disturbed, it's going to disturb our tectorial membrane there, number 2. Tectorial membranes are going to slam down on top of those hair cells, and then we can see that basilar membrane that each of those hair cells are sitting on top of. Those pressure waves, you can see after they've caused the hair cells to pick up sound, those pressure waves continue to move down into this tympanic duct. And the way they get out of that tympanic duct is through the round window. So out through the round window, and then those pressure waves balance out. We said the eustachian's tube job, its job was to make sure the pressure stayed equal on either side of this eardrum, that tympanic membrane. Okay, so let's see if I can make that point with the fact that I said if it's really high, screechy, like trebly notes, these are going to move across this cochlear duct sooner than a low, bassy note. That's what this slide is attempting to show. It's attempting to show that this bending cochlear duct here is more sensitive to high-pitched tones early on, and the further you get into this cochlea, it's going to be picking up lower, kind of bassier tones. And that kind of makes sense. If we had somebody across the room here playing some like trebly music, we probably wouldn't hear that. It wouldn't go through the wall as easy as if they were playing lower, bassier notes. That goes through deeper into the cochlea, just like it would go through the wall easier. One way of kind of thinking about that, if we take this, again, these 3 pictures are showing the same thing. If I take this basilar membrane that all the hair cells are sitting on top of, and we think of one end of this basilar membrane as being sensitive to high-pitched notes, and the other end of this basilar membrane being sensitive to low-pitched notes, it's almost like we have an inverted piano, where if we were to take this coiled organ and stretch it out flat, here's our coiled organ, and we're stretching it out flat, we would see the high-pitched notes are going to be picked up closer to those oval windows and round windows. Low-pitched notes, again, the opposite of the ways the keys are laid out on a piano, where you got your low end over here and your high end up on this side. It's the opposite for the basilar membrane. And here's a little video that shows sound waves, whether they're high pitch, low pitch, like these low frequency notes versus high frequency notes, they vibrate at different places along this basilar membrane. High pitch always towards the beginning, and then low pitch always towards the end. Kind of extreme, but I like that it shows that when you hear complex things like this, that you've got multiple parts of that basilar membrane that vibrate simultaneously, because they each are sensitive to different frequencies of sounds. also how certain, like you can become tone deaf to certain frequencies. And if you hear, one frequency more than others, then the hair cells in that particular region can get worn out. Pitch is just referring to the wave frequency. So if you look at the wave frequency of these first two sets of waves that we have, you can see the frequency between those peaks are the same. So this is going to be the same note, just at different volumes. The volume doesn't have anything to do with the frequency of waves. The volume has to do with the amplitude of those waves. So A and B, these are the same note, the same pitch, but it's just at different volumes. This is at a higher volume, this is at a lower volume. Between C and D, you can see the wavelengths between C and D are the same. So these are both, since the wavelengths are more drawn out, separate from one another, then we can see them compressed together here in A and B. These wavelengths that are more drawn out are going to be a lower pitch note. This would be a very loud, low pitch note, and this would be a very quiet, low pitch note. So the pitch has to do with the frequency of wavelengths, and then the volume has to do with the amplitude of those wavelengths. Same thing that we're seeing here. We can see if the wavelengths are stretched out versus compressed wavelengths are high pitch notes. Stretched out wavelengths are low pitch notes. You can see the amplitude of the waves here that's the same amplitude. So these are the same in terms of their volume, but they're different in terms of their pitch. Our low pitch versus high pitch tone. Down here we can see the wavelengths are the same. So these are both going to be the same note, but they have different amplitudes. So the loudness is going to be different. Loud versus quiet. And then down here, this last one is called timbre. And this is just something that's referred to as maybe the quality of the sound waves. A better way of describing this timbre is it's the overtones that you hear when somebody is making sounds. So the little blue overtones that we can see, this would be, my analogy would be if I hear my mom call me on the phone, I can pick out my mom's voice from anybody else's, and that's because I can detect, I'm aware of her certain overtones that she uses, or even my weird voice whenever I'm saying things. My voice is a little different than other people's voice, and our own voices are indications of our own little overtones that we put on the words that we use. So being able to recognize somebody's voice is you're recognizing those little personal overtones that they put on the sounds that they make. But all that this was really pointing out is that those sensory neurons, as we've kind of drawn them here on the board, once those are picking up the vibrations of that tectorial membrane, the sound is going to be actually sent up always through the thalamus. This is the router of the sensory information that comes into the brain. But sound waves are going to be sent over to the temporal lobe. If we've mapped out these pathways into the brain, we said for vision, visual stimuli are processed in the occipital lobe, the back part of the brain. If you've ever stimulated the back part of your brain by accident, maybe like falling backwards and hitting the back of your head, sometimes that will cause you to see these stars. You've stimulated these neurons in the occipital lobe whose job it is to perceive images, and accidentally stimulating those could cause you to see these stars for a second. So occipital lobe for visual stimuli. If we're thinking about auditory stimuli, sounds are going to be sent to the temporal lobe. So temporal lobe for processing auditory sounds.